Our next speaker is Dr. SHAKEEL MODAK. He is the chief of the neuroblastoma services at Sloan Kettering and professor of pediatrics at Weill Cornell Medical College. He trained in pediatric hematology oncology in India, England, and MSK. He has been involved in neuroblastoma research since 1997, with research interests obviously in neuroblastoma biology and immunotherapy. He has served on several foundations and governmental agencies, and he will be presenting some updates on Sloan trials and the hTERT. Dr. MODAK.
Pat, thank you so much, and such a joy to see so many people, and bittersweet to see parents whose kids have passed away, and always hard to see that. But there's a lot going on in neuroblastoma, and there are reasons to be positive, both now and down the line. I don't think I'm going to talk much about the hTERT business, but generally give an update on where we are today and where a few things going forward. These are my disclosures. What am I pointing at? All right, great. So I want to talk about a few disparate things. One is the state of induction chemotherapy, and 9 and 10 it should read. Now when I don't need it, yeah. Naxitamab, we have been using for a long time, but now finally we've gotten out to publish our experience. Many of you know about the vaccine. I want to give you an update on that and where we are going with it. MFBG, again, another exciting development with pros and cons. And want to update you on about that, and a couple of new initiatives that have clinical impact. So kind of a disparate agenda. So if you look at induction chemotherapy, we know that getting patients into complete remission is important, because if we don't get patients to complete remission, their outcomes are not as good as those patients who get into complete remission. And thus far, if we look at all the historical data, the complete response rate to induction chemotherapy has been about, in general, about 50% at best. So definitely a place where we can do better. And we have done better. Both our group and St. Jude's group, and now Children's Oncology Group has launched a new trial, as many of you know, about adding immunotherapy earlier. So what have we learned so far about induction chemotherapy? We know that we do need a certain amount of chemotherapy in the beginning. Our current chemotherapy is more effective than it was 30 years ago, but still not good enough, because if you're only getting 50% of our patients to remission, we need to do something different. We know that too much chemotherapy is also not good. For example, now we've reduced our number of cycles that we give in the beginning to only 4. It used to be 7 at one point. We know that many of our patients use hearing aids, and that's because of cisplatin. And as our current studies remove cisplatin altogether from the regimen in an effort to avoid this toxicity. And again, as I'd mentioned, that achieving complete remission is our goal for this bit of the journey of neuroblastoma. So we completed our N9 pilot study where we took 15 patients. We treated them with 4 cycles of chemotherapy. So cyclophosphamide topotecan, ICE, cyclophosphamide doxorubicin, and then cyclophosphamide topotecan. And we do collect stem cells, but not for transplant. But if, for example, if patients need MIBG therapy later on, we have those in our back pocket. And we treated these 15 patients, and we were able to achieve resection in all of them. So that means adding the carboplatin instead of the cisplatin at least did not affect our ability to do good surgery. We were also able to collect the number of stem cells that we wanted. Again, that was reassuring that we were able to meet these two goals. But the response to this treatment was the complete remissions were achieved only in 6 out of the 17 patients, although another 5 patients achieved a partial remission. So this is very much in keeping with what chemotherapy did or used to look like before the advent of antibody. But we were not disappointed because we know that some patients are not going to respond. And those patients who did not respond or did not get into CR, when we treated them with antibody alone or antibody plus GM-CSF alone without any more chemotherapy, we were able to achieve 7 more patients going into complete remission. That means a total of 13 patients got into complete remission. So what does this whole approach allows us to use antibody earlier on, and we don't keep on doing chemotherapy if it's not useful. Now, whether adding antibody even earlier on, that is after the second cycle, which COG is testing now, is that going to be even better? That question should be answered soon. But when we launched this study, we really wanted to move away from the cisplatin, so we could not make too many changes. But at the end of this, and looking at all the results over the last 2 or 3 decades, in a nutshell, we think that some patients are not going to respond to chemotherapy, and these patients don't deserve to keep on being exposed to chemotherapy. But if they have bone and bone marrow disease alone, they're going to respond very well to antibody treatment. Then another group of patients are going to have a very good response to chemotherapy. And in this patient, you could argue that they need more chemotherapy with transplant, or you can also argue that they should get no more chemotherapy because they've already achieved remission. So for the first group of patients, we expect them to have a very good response to antibody plus GM-CSF. The second group of patients, we think that they don't need any more chemotherapy and antibody is enough to prevent relapse. And then a third group of patients have a partial response to chemotherapy. And maybe this group of patients needs some more chemotherapy. For example, they could be treated with chemoimmunotherapy. So this is kind of the philosophy that we've come to, and we are testing it now in this N10 study, which will basically look like this. Now there have been about 7 patients enrolled. So chemotherapy for 4 cycles. If complete remission, we do antibody with naxitamab without any transplant. If patients get stable disease in bones and bone marrow only, we give 2 cycles of immunotherapy with naxitamab plus GM. And if they're going to remission, we'll do 5 cycles of antibody after that. Those patients who achieve a partial remission will get chemoimmunotherapy. So this is kind of a modular approach. It's easier to do in a single center. So how do we translate into multi-centers will be a question. Because when you have a single center, you can analyze the response in a timely manner. But we look upon this as a pilot, and there's also a concurrent study going on in COG will also help us determine whether adding antibody early on is good, which we think it is, but we need to prove it. And why are we confident? Because St. Jude's also reported a good complete response rate with adding antibody early. And when we went and looked at our N7 data, where we had reported a 79% CR rate, we think that was high because we also added antibody early on after cycle 5. So putting everything together, I'm enthusiastic about adding antibody earlier on. And we'll kind of figure this out and see how much of a difference we made long term. So that's where we are with our induction protocol right now. And to pivot and talk about where we are with naxitamab. So naxitamab is a humanized anti-GD2 antibody that was based on the 3F8 platform, which was a mouse antibody. And recently we've published a few papers on naxitamab, which I kind of wanted to touch upon. So when we look at bone and bone marrow disease alone in patients who have not responded, and we call this group primary refractory group, in 32 patients, we were able to get them into very robust remission without any chemotherapy. And in another study where there were relapse and refractory patients, there was a 50% response, again, without any chemotherapy. So this is quite encouraging, and we see these responses even after 2 cycles of naxitamab plus GM-CSF. Then in the old days, we used to get patients into remission, and then we would give them mouse 3F8. Now we've moved to naxitamab plus GM-CSF. So how do these patients do? And again, our progression-free survival at 3 years is 75%, and our overall survival is about 85%. That is, when we use naxitamab to consolidate remission without transplant, almost large majority of these patients did not have transplant. We were able to see the same, if not better, outcomes as we did when we used mouse antibody. So again, this corroborates, or at least gives us confidence that we will be continuing this treatment without transplant, at least in the near future. And when this was also looked at by Dr. Mora in Barcelona, and again, he was able to reproduce our data. That is, in patients without transplant, he was able to achieve good event-free survival for a long period of time. And another update is on using antibody plus chemotherapy. And both dinutuximab and naxitamab have been used in combination with chemotherapy, and we do see similar response rates. And so that even in patients who had irinotecan and temozolomide before, patients who had one antibody or the other before, when we have relapsed disease, we do see good, at least the best responses that we have for second-line treatment. Our trial hasn't been published yet, but it should be published soon. And Dr. Mora has also reported similar data, especially if hits was started early on rather than later on in the process. As many of your kids have experienced, naxitamab can be a challenge to tolerate, and we are constantly trying to figure out ways of making it easier for patients. As you know, naxitamab is given outpatient. So there are 3 or 4 things that have made this experience better. One is we've improved our pre-medication. Now we can use ketamine outpatient, even as a continuous infusion. We found that using methadone up front also reduces the pain. And both Dr. Mora and us now use what's called a step-up method. We infuse naxitamab slowly for 5 minutes, increase it every 5 minutes, and that seems to saturate the sympathetic receptors, and we see less of low blood pressure when we use that method. And you can also say if you give it over a longer period of time, you might have more pain. But we also use ketamine in conjunction with the step-up method to try to reduce some of the side effects. Bad side effects of urticaria, severe rashes. We've used another drug called omalizumab. And overall, I think as more people use naxitamab, other centers have reported on their experience regarding how they've been able to tweak the naxitamab to do this safely. So now I wanted to talk about vaccine. So when a patient relapses, we try to get the patient back into remission. And using whatever tools that we have at our disposal, this could be second-line chemotherapy, chemoimmunotherapy, MIBG therapy, or say even genomically guided therapy. As long as we can get the patient in remission, that's our goal. And once the patient is in remission, we reconsolidate with naxitamab. And then the question arose, can we improve outcomes by adding the vaccine? So that's where we first tested the vaccine, which is a shot that's given now. It's given for 3 years, 7 injections the first year, 2 in the second year, and 1 in the third year. And the goal was to see if this curve, which you see, the survival curve, was our survival curves before the vaccine. In the old days, 15, 20 years ago, we would get patients with remission and treat them with naxitamab alone or 3F8 alone. And we want to improve that with the vaccine. Can we improve on our outcomes? So one thing we have learned that if patients respond well to vaccine, their progression-free survival and overall survival is much better. So our goal is in the vaccine is to improve this anti-GD2 titer. We want to, if this makes so much of a difference, we want to make it better. And that's what the goal of the current vaccine trial is, to improve the anti-GD2 titer. So we learned that even patients who had previously relapsed, we were able to maintain durable remissions. Patients who had 1 relapse versus 2 or 3 or 4 relapse also mattered. So if you had only 1 relapse, your outcome was better, which is logical. And we didn't really see any side effects of vaccine. So when we kind of looked at this data, we said, let's try to make the anti-GD2 titer better. So the first thing we did was to see when the glucan, which is a drug that's taken by mouth to make the vaccine work better, should be added, and we think that it should be added earlier on rather than later on. And then we now are asking, can we do even better? And so we've added the GM-CSF as another adjuvant. So the current vaccine trial has 2 groups. One is the standard vaccine, and the other is vaccine plus GM-CSF. And we accrued quite quickly on this trial, and I'm hoping by the end of next year we would have finished accrual. And if the GM-CSF arm does better, then that will be the standard. But importantly is, what do we do with all of this? How do we function as a single center? And this is a question I don't have answers to. So say if we go to the FDA with our data, the FDA will tell us, you're a single center, better do it, you have to do it in multiple centers. And in order to do that, we need pharma involvement. That has not materialized thus far. Another thing that we discovered is that a molecule called dectin dictates how patients deal with the vaccine. If you have a good dectin, you're going to do better. But some adjuvants, newer types of immune stimulants, may overcome the negative aspects of dectin. And so that's why the GM-CSF was added. But in order for this to be approved, it's likely that we'll have to revamp some of the components of the vaccine because they are quite archaic in some ways. Whether we should do that or not is another question. And ideally, if we had pharma support, we would do this in a multi-center study, and that is our goal. So the way I look at it is that we finish our current trials in 2 years, say this is the ideal type of vaccine, and hopefully by that time, we might have some pharma support, and hopefully this could be a multi-center trial. So I'm kind of speculating here, but this kind of is our long-term or intermediate-term plan for the vaccine. MFBG was also first pioneered at MSK. It's with Dr. Pandit Taskar as the PI. And essentially, when we look at MIBG scans, we see very hazy images. But nuclear medicine would like to have much clearer images, maybe even count each dot and see how many metastases there are. When we look at MIBG scans, we see a much more blurred picture, and it becomes difficult to quantify. So we tried to changed the MIBG to a PET scan. So we removed the iodine of MIBG and replaced it with fluorine, so MFBG. And now we get much better images, and we don't need thyroid blockage because the fluorine doesn't affect the thyroid, and we can image on the same day. And now this has been also done in other centers around the world, like China and in Europe, in Denmark and the Netherlands. So in a nutshell, this is a very good scan in my view, in the sense that it makes the nuclear medicine doc's life easier. But you could also argue that this is a more sensitive scan. So if you pick up more dots, are we going to be more stressed out? Or are we going to detect relapse earlier? And to do all that, there is a phase 3 study that was done by pharma, which hasn't been completely analyzed yet. But in our own study, in 37 patients, we actually detected 200 more spots than the MIBG could. And these spots were actually real. That means patients, as we followed them, did relapse in the areas we picked up. So my take home, or at least my interpretation of this, is that this is a test that can actually pick up disease much better than MIBG. But how do we use it in our practice is a question, because if we use a sensitive test, we will have to change all the criteria we have now of response assessment. And that probably should be done, but how to do it it'll probably take some time before this becomes a standard test. And again, we need to make sure that we are not picking up false positives, because if we pick up a false positive, then we'll put our kids through more scans and more anesthesia. We are kind of getting there, but we are not there yet. At this meeting, we have a poster on our study, and then tomorrow or day after, I'll be talking about the pharma study, where they are. And eventually, this actually might even be a cheaper imaging agent, because fluorine is much cheaper to get than iodine, and especially in the developing world where iodine 123 is not available. This actually may not necessarily be a more expensive way of doing things. And lastly, I want to talk about some other topics of interest to both us as physicians and you as parents and patients. One big issue is, can we detect minimal residual disease earlier? Say, I finished antibody treatment and I'm in remission. Can I predict who's going to relapse? Because if we have such a test, then perhaps we can do something about it. We can do, say, more cycles of antibody, or we could do DFMO, or we could do a vaccine, right? So if the scans are clear, there is a 35% chance of relapse still. And can we make a more sophisticated test, such as circulating DNA? And currently, the circulating DNA is not there yet. That means we can't use ctDNA as a substitute for scans. It's definitely less sensitive and specific as it stands currently. But there's a lot of work going on here. So Rachel Hughes is a fellow in our at MSK who has looked at a new way of detecting ctDNA, specifically for the purpose of MRD. And we recently got some preliminary data validating this test in patients who have disease. And now we are going to use this test in our vaccine patients to see if we can predict who is going to relapse and who is not. And if we can answer such a difficult question, I think this could be very useful in our day-to-day management. We have launched a bigger program now of looking at circulating DNA like other centers have, not only for MRD, but to guide our treatment, find new therapies, and that's being spearheaded by Dr. Angelini, who's joined our group. And finally, we are looking at other ways of looking at DNA sequencing. What we do now currently, it's called short reads of DNA. That DNA is chopped up, and each fragment is sequenced and analyzed. But if we can do long reads, That is, we won't miss something in the middle which has been chopped up. And so that's a new approach, has been done for other diseases, and the technology, which was much more expensive, is now available. So that's another initiative that we have. And finally, Dr. Iglesias, based on data generated out of the Maris lab, has launched a study on this inhibitor of a molecule called PLK4, which is seen associated with this another molecule called TRIM37, which is present in neuroblastoma. And that study currently is open to adults or patients above the age of 12 via a company. So we are still at the mercy of the company. We have enrolled 7 or 8 patients maybe on this. And I think it's too early to say whether this is useful or not. And also we'll need a pharma support. And in order to get pharma support, typically it means that this drug has to be also beneficial for some other diseases. So all that is still up in the air. A pediatric study is supposed to launch this year, but I don't have any further updates on that. That is in patients who are less than 12, I mean. So I want to thank Pat for she's done such a fantastic job over the years, decades. And I think it's kind of, I have to admire all the strength and stamina that she has in keeping this forum going. Hopefully it's of use to you guys, and always found that my parents are really up to date with everything, sometimes more than I am, and I think a lot has to do with the opportunities this type of forum provides. So thank you.
Thank you, Dr. Modak. Does anyone have any questions? Got a question there? 2 questions? Wait, you got somebody behind you, Frank.
Also, I mean, I have my email address, so if you can feel free to text me, email me.
Mm-hmm. On the new scan that's being contemplated, what other centers would be capable of doing that if it?
It's an easier scan to do.
Would they be able to adjust, adapt right away?
Yes, because the PET scanner, I don't know if you're aware, but now there's a new PET scanner. It's been used in China and some parts of Europe, where you can do a PET MRI in minutes.
Oh.
So you don't even need anesthesia. And that's now, we have that also, and other few centers. So that can really reduce the risk reduce all the anesthesias that we need. But because FDG PET is used by every center,
yes,
it's easier for nuclear medicine docs to analyze that test rather than MIBG. Mm-hmm. Most places with 123 MIBG, the know-how is important. But paradoxically, for the PET, it's very routine. So yes, the answer is this will be an easier test for nuclear medicine docs. But whether these spots are truly positive and we're not picking up false positives, that's the danger. Because then if these are false positives, we might end up doing more scans on a patient which they don't deserve.
Yeah. Thank you, Doctor.
Sure.
Another question?
Hi, I wanted to hear if you all have an update on the microbiome study.
Yeah, it is about to be published. This is the last set of experiments. I didn't put it in here. So the microbiome means that we take the stool samples from patients, we analyze the bacteria that are there, and we try to correlate the bacterial genetics with the immune response. So this has become quite, it's a fast-moving field. Even in the field of checkpoint inhibitors, that's the immunotherapy in adults, where people get pembrolizumab or nivolumab, and the question was asked whether a fecal transplant could, because the microbiome, there are 1 trillion bacteria within our gut, and that could, we know that some things like nutrition, our metabolic syndrome like diabetes and obesity are definitely impacted by this. Now, neuroblastoma is a rare disease, so the number of patients are small, but the data points that we get from the analysis are huge. we get a lot of data for a rare disease. So it is still possible to use that data to help us. There are two ways this could help us. One is if there's microbiome X or microbiome Y, X does worse than Y, then we kind of can explain why some of our patients relapse. But I don't think that's going to be the outcome because there's so many different things that affect our patients. Mechanism amplification, how much chemotherapy they got, ATRX deletions, hTERT, et cetera, et cetera. So this is going to be one part of that whole kind of jigsaw. But the other question is more relevant for us and our patients because can we change the microbiome, right? Can we change the microbiome to become a positive microbiome? So therefore use a probiotic, not in the traditional sense of the over-the-counter probiotics, but a much more targeted, specific probiotic. That is where the money is, and that's something that we are working on. There's a lot of issues with microbiome because our microbiome on the East Coast might be different from the West Coast. Microbiome from the US might be different from Bangladesh, for example. So all those questions also have to be answered, but our goal is to find a probiotic that can help with immunotherapy, and that's what the fecal transplants were done, but those have not been validated yet. So there are a lot of data points to kind of decipher. We have found some bacteria that are associated with better outcomes in vaccine patients than others. And I saw a paper, and we also looked at this from China, that's looking at microbiome in patients who are getting antibody. But this will not be like a smoking gun, but I think if we can change the microbiome to some degree to make things better, that would be how we as someone who's in the trenches and parents can benefit from that. But still a ways to go, a little bit more, either positive or negative, very soon we'll know.
Okay, thank you.
Thanks.
Okay, well, a couple more questions and then we'll be done.
Clarification, please. Your vaccine, is it more just for relapsed patients, or is it as a maintenance?
To prevent a remission, prevent a relapse. Right now, the primary objective of the vaccine is not really to study the relapse rates and long-term survival is the secondary objective. What we want to do, what we are trying to do now is to find the ideal vaccine. So we've tweaked the 1, 2, or 3 components. Now we're tweaking a third component because we don't know what's the best vaccine. Now, the secondary goal is to see if we are making a difference. So for patients who have not relapsed, say my kid goes into remission, I get antibody treatment, I finish the antibody treatment, and the scans are clean. At that point, the chances of another relapse or chances of relapse are about 30%. Right? So if at this point you start the clock, there's a 70% chance of cure. So anything we do in that point of the journey has to bump up that outcome to, say, 90%, for example. DFMO was a drug that was approved in this landscape. And that was approved because the group of patients was very tightly controlled. So it was able to be studied in those patients who were transplanted, then dinutuximab and Accutane, and then the DFMO. We don't have that luxury of defining that group of patients for vaccines. So in my opinion, we will need to do a randomized study. That means treat half the patients with vaccine, another half with something else, and see which group does better. That is for those patients who never relapsed. So in order for us to say with complete confidence that vaccine is good for that group of patients, in my opinion, we need to do that. Now, there's another group of patients who are the second remission. That means patients relapsed, they got treatment X, Y, and Z, chemoimmunotherapy, MIB therapy, high-dose chemotherapy, and went to remission. A textbook will tell you that this patient will invariably relapse, which we don't quite buy into. But if we can show in this group of patients that by doing vaccine, we have done better, then in my mind, that validates what we're doing. So in that group of patients, our numbers do seem to be better than before. But again, to do this in a way that can be approved by the FDA, we'll need some more work. But whenever a patient asks me, should I do something after antibody, that has to be taken in the context of already having a 70% chance of cure. So I never tell a patient do vaccine there necessarily, especially if you have to travel and so on, right? And there is a DFMO. If you've gone through the dinutuximab, then the DFMO has been approved. Patients who've relapsed, then gone back into remission, it's very reasonable to consider vaccine. That's kind of my take on it. Is that the question? Yeah.
Okay, Josh, go ahead.
Thank you for coming. Correct me if I'm wrong, but we know that the titers are a big indicator if the vaccine will be successful or not. Have you guys looked at how robust a patient's maybe immune system is going into that to be a potential indicator? I'm kind of thinking about transplant versus not, and how may that impact someone's immune system going into that?
The immune system, when we use that word, it's a non-specific, and there are hundreds of components of the immune system, right? So by the time we start the vaccine, you're already about 6 months out of transplant, or 6 months from the end of high-dose chemotherapy. So a standard immune assessment would indicate that your immune system has recovered, because that's the time point when we stop Bactrim, for example, right? So the global immune system has recovered enough to mount an immune response by the time we start the vaccine. Now, which component of that immune system led to a titer? That is going to be very difficult to tease out. And whether the titer is the only thing that we need to measure is also can be a question, because this field is moving so fast. So a global recovery of the immune system has happened around 6 months. But all the details as to which of these led to a titer and which did not, we don't know. And the second question was whether transplant versus no transplant. It didn't really make a difference in the titer. Yeah.
Thank you.
Sure.
My question is about the MFBG. That concept term was introduced to me 2 years ago at the same conference, and here we are still talking about it. You mentioned a couple different times, like, it's something that's still in the works. How can we as parents help advocate to help oncologists, institutions? I mean, do we reach out to our Congress
So
people?
now it's a company that's taken over. So for the last 2 years, the last study, we cannot do any study anymore. None of the academic institutions, because there's a company that took it over and conducted a study and completed accrual. And they need to persuade the FDA that this is good.
All right.
So they've already gone to the FDA, and I can't say any more because I don't know any more details.
And then
Now, if it is approved by the FDA, then it would be a question. It'll come back to our, as pediatric oncologists looking after neuroblastoma, back into our court, because then we'll have to design new response criteria for assessing response to say this is remission versus no remission.
Okay, so with the scanning of the MFPG, you mentioned you don't want false positives. Have you seen that?
We've just completed a big study on, there are only 37 patients, but because we counted each and every dot, right, we saw 229 extra dots. And in our experience, each and every of those dots is real. So as a single institution, when we did our study, we think that these are true positives, not false positives.
Okay.
But that also has to be validated by in a multi-center study, because other eyes have to look at it. We had only one nuclear med doc look at it, and she was also part of the study. So whenever that happens, there's a bias. On the other hand, these are all our patients, and we scanned them repeatedly, so we could say with some confidence. But if the multi-center study confirms this, then this is a true positive. Then we could also use this test as early detection. So that needs to be confirmed.
And that would ultimately replace the MIBG. And
If everything was ideal, yes, it should replace it. This is my personal opinion.
Yes, your
okay?
opinion.
But for example, 30 years ago, when I was a trainee, we were using 131 MIBG, 131 iodine MIBG. Nobody would do it now because it's very poor quality imaging. So this is, but it took 20 years for the new MIBG to come in because people needed to be trained, people didn't have the right scanner, et cetera, et cetera. But now the nuclear medicine field is really geared towards it because they have the scanners more accessible than for 123. So it should not take 20 years.
Yeah. Well, I just feel like even with the MIBG, different things light up in the body all over that don't really pertain to neuroblastoma specific. Like your lymph nodes will, light up, saliva glands light up.
No, salivary
so-
glands and lacrimal glands will light up on both because
Both,
that's
okay.
that's taken up because there's a lot of sympathetic nerves there. But we don't mind that because we can differentiate those things very clearly with the MFBG.
Right.
The MIBG, you don't know, is that a node, is that a salivary gland, lacrimal gland? It's much more difficult to separate out. But they will continue to light up, but they'll be much sharper,
And
and the
clearer.
nuclear med docs can tease that out much better.
Okay. And I just want to thank you for all you do. I
Thank
see
you.
your
Thank
name
you. Thank you.
everywhere on Facebook and a lot of our support groups.
It's
so
dangerous.
Thank you.
But
And
thank
thank
you
you,
for all your thoughtfulness, and I always appreciate you, everyone's ideas and thoughts.
Okay.
Thank you.
Thank you, Dr. Modak.